Hello everyone, and welcome to the fourth live session within the third week of the first International Electronic Conference on Genes, Theoretical and Applied Genomics. This conference has been organized by MDPI and MDPI Genes Journal and is being chaired by Dr. Aureliano Bombarelli from the University of Milan in Italy. As always, I would like to say thank you to our chair and the conference committee for making this conference possible, and all the participants for joining us today. A reminder that at the end of until the end of the month, everyone can read and comment all the author submissions presented for the conference, and also enjoy some recorded and live sessions. Please look at our webpage for more updates and register for free for the last live session that we have next week. I would like to remind everyone that this webinar is being recorded and that we have a YouTube live streaming today. I will be sharing the link through the chat in a minute. The recording will be posted on the conference webpage for everyone to watch it as soon as possible. Also, at the end of the session, we will have a QA session. So if you have any questions, please feel free to post them in the QA box at any time. If you would like to ask your questions on live, use the raise hand option, and I will upgrade you as a panelist for a moment, letting you have your camera and your microphone on for asking your question. So if anyone needs anything during the session, please write me using the chat option. The speaker for this session is Stahl Rommel. Sarah is a research scientist in the microbiology laboratory from the NASA Johnson Space Center. She has served as a project scientist for many analog and space flight investigations, including the biomolecule sequencer flight experiment that demonstrated the first DNA sequencing aboard the International Space Station. Sarah has overseen the implementation of a culture-independent direct swab-to-sequencer method in many extreme environments, including the International Space Station. Today, she will present her talk entitled Sequencing the Extremes and In-Betweens, Rapid Swab-to-Sequencer Microbial Profiling from the Seafloor to the Space Station. I will hand out the microphone to her now. Thank you, Sarah, for accepting to participate in this session, and everyone enjoy.
All right, well, thank you so much for having me. Let me go ahead and get my presentation pulled up. All right, so I'm really excited to be here with you guys today to get to talk to you about the work that we've been doing with my group at NASA to develop this rapid swab-to-sequencing microbial profiling method that allows us to detect microbes in extreme environments. So I wanted to start out with just this high-level overview of NASA's current manned spaceflight mission plan. So currently, NASA sends astronauts to the International Space Station, which is in low Earth orbit. And the crew members live and work there for about 6 months to a year, and they conduct really valuable experiments that help us learn more about living and working in the spaceflight environment. And that information will be really valuable as we move towards these deep space missions. So the current objective is to return humans to the surface of the moon by 2024. And we're also excited to be working on developing this new gateway habitat, which will be really similar to the International Space Station, but will orbit the moon, and it will allow crew members to have access now to new areas of the moon that we haven't previously explored. So we're really excited about improving our abilities to do more exploration. The gateway habitat could also be used to build a Mars transit vehicle to then take crew further on to Mars. So with these deep space missions in mind, we really need to focus on how the crew can have better crew autonomy and reduce a lot of the reliance that we currently have on the ground with the ISS. So today I'm going to focus specifically on the work that we've been working with the ISS to help us work on better microbial monitoring in the future. So as a spaceflight microbiologist, my main focus is really on crew health. So I want to make sure that the crew is staying safe and that everything in the spaceflight environment is optimal for their best performance during their spaceflight mission. We're also really interested in how the vehicle is performing microbially, and also all the life support systems, and making sure that everything is functioning optimally. Part of that is really understanding how microorganisms respond to the spaceflight environment. There's been a lot of research by Dr. Cheryl Nickerson to show that salmonella actually can become more virulent in the spaceflight environment, which could be really harmful to the crew members. So it's really studying different microbes and how they respond. And also, finally, we do a lot of work on testing new technologies and how we can increase our ability to do more microbial risk assessments. So the main microbial monitoring that we do is monitoring the air, surface, waters, and food. So at NASA, we have a specific acceptability limits criteria where we do pre-flight and in-flight monitoring. So we sample the vehicle surfaces and air before launching, and then we also sample all of like the hardware and cargo bags that go up to the space station, as well as food. So if none of these items, if they fail our acceptability limits, then they will not be able to fly until they've been remediated and able to fit within our limits. And those limits are specifically based on how much microbes are there and what is it. We also do microbial monitoring in flight, and this happens about quarterly, and the crew members will collect surface samples, air samples, and water samples. So for the surface and air, they collect the bacteria onto a TSA media, and for the fungus, they put it onto a sabdextrose media. And they let it grow for 5 to 7 days, and then can do a preliminary plate count and report that to the ground to say, hey, there's approximately 10 colonies on this plate, but we don't know what it is until it can get returned to the ground, which can take about 30 days to get it to the ground and be able to identify what is there. Same thing with water. They collect a water sample from the potable water dispenser, and they can put it onto this microbial capture device that you can see in the right hand corner. And so they filter the water onto that and then add some R3 media. And those little blue dots are actually colonies. So there's a dye that turns the colonies blue, so they're easy to count. So we can get an estimation of how much microbes are in the water, but again, we don't know what it is. The only way that we can do any kind of detection of what could be in the water on board is through this coliform detection. So you can see that in the bottom right corner. The liquid will turn yellow if it's negative and purple if it's positive. So the purple would mean that there could be some kind of enteric microorganism that could make the crew sick. So then that would trigger some remediations. But as I said earlier, we don't know what it is specifically until we get things back to the ground, which can take a lot of time for us then to recommend a remediation and expand the time that the crew are potentially exposed to a risk. Luckily, the ISS is actually a very clean environment. It's about the size of a 5-bedroom house. And I love this figure here on the right side because it shows the bars are showing 5 or 6 different houses, an average home. And the lines are showing the ISS detection limits. So in blue, it's bacteria, and red, it's fungus. And you can see that most average homes are at or above the limits of the ISS, and the ISS is usually below those limits unless we have a rare outbreak occurrence. So the ISS is a fairly clean environment. But that being said, it has been constantly habited for the last 20 years, and the crew will often find weird things going on, and they'll send us a picture and say, hey, what is this? Should I be concerned? Is this biological? What should I do? And we'll just say like, hey, take a better picture, take a sample of it and send it back to us. But depending on when this outbreak occurs and when a vehicle's returning, it could be several months before we can get an answer. So we think that wouldn't it be great if there's a way that we could bring the lab to the crew and let them kind of do more of these investigations themselves? And speaking of technology, we've seen the technology of spaceflight change over the years. Over the last 60 years, we've progressed into different focus of spaceflight. And as we move into these more deep space missions, everything is changing again. So our microbiology has actually relied on culture-based methods for these entire 60 years. So as we move towards deep space, we can't rely on sample return. How can we still continue to do our microbial monitoring? So this brings me to 2016. I like to call it the dawn of the molecular space age. So this is when everything has changed for doing molecular microbiology in spaceflight. So there were two really important payloads that occurred. So in April 2016, there was the Genes in Space 1 payload, which was the first molecular biology experiment done in space. It was doing a polymerase chain reaction using the mini PCR machine, which you can see the astronaut holding here, from mini PCR Bio. And Genes in Space is actually a student program. It's very cool. It's for middle school and high school students to submit proposals to get to do molecular biology in space. So they have really done a lot in expanding molecular capabilities on the space station. The second payload was in August 2016, and that was the biomolecule sequencer project. And that was actually my first spaceflight experiment. And we validated that DNA sequencing can perform in space. And that was using the MinION sequencer from Oxford Nanopore Technologies. So just a little bit more information on that. If you're not familiar with the MinION sequencer, it's a extremely portable sequencer that can fit in the palm of your hand. And it works completely different than standard laboratory sequencers by using this flow cell that you can see in the middle there. That flow cell contains about 2,000 nanopore proteins. And when the DNA gets pulled through that nanopore, it blocks the current. And the current change can then get decoded by the software to tell you what kind of bases you're reading. So this technology makes it extremely portable, and a lot of researchers have been using it to detect Ebola and Zika and COVID out in the field. So currently, this is the only sequencer that is compatible with the spaceflight environment. So we did this experiment to validate that it works in space. So basically what I did is I prepared samples on the ground and froze them, and then launched it to the ISS, where Dr. Kate Rubens thawed it and loaded it into the sequencer. And then I thawed it and loaded it on the ground. And then we were able to compare those results and show that the sequencer performs just as well in the spaceflight environment as it does on the ground. And this was really a huge jumping off point for increasing molecular capabilities on the space station. But what was the point of having a sequencer if we don't have a way to actually prepare our samples in the spaceflight environment to make everything be able to be done in situ? So that's where we started with this goal to create a complete swab-to-sequencer culture-independent method that could be run completely on the International Space Station. So we set out the crew member would collect a swab from a surface. They could extract the DNA from that swab and prepare it for sequencing, doing a PCR reaction and all the library prep necessary to load it and run it on the sequencer. But in order to do that, the first thing I started to take into consideration is how do you even do sample preparation in space? So it's not like working in a normal laboratory. You have a lot of different requirements and regulations that we have to abide by. So our number one concern is crew safety. We really want to make sure that we're meeting the toxicology and the flammability limits. We have to make sure we're not putting the crew in any danger in what the reagents we're giving them, or if there's any like sharp edges on our hardware. We need to be cognizant of the space that we take up. Part of the reason this has been so successful is that we have extremely portable hardware that can be used in extreme environments. We really needed to consider microgravity. How would we be able to secure things? And you can see from this top picture here that we use a lot of Velcro and tape to just hold things down. I love that the crew members just like flip the pipette upside down and put it onto a piece of tape, and it just kind of floats around there. The other things to really take into consideration are crew time. They're often, currently, I think we have about 7 crew members on the space station right now, but right before the new vehicle arrived, we only had 3. So we really need to make sure that we're not wasting a lot of their time. They're really busy doing a lot of different experiments. We have limited temperature storage, so we only have ambient 4 degrees Celsius and minus 90 degrees Celsius. And then finally, vehicle transportation. So we can only resupply items every so often. So with all of those ISS constraints in mind, and also knowing that the ISS is a very low biomass environment, we set out to develop a method for our specific goal. So the first thing that I was really digging into is swab type. So I looked at some foam swabs, nylon flock swabs, several different types of polyester swabs before identifying one that was able to have the highest microbial yields. We had to also get creative with our buffer selection. So we can't use the typical ethanols that you would be using in kits because of the flammability. So we had to come up with some substitutions. For DNA extractions, the first thing that most scientists always do when they have a new experiment is just go and grab a kit. But we quickly found that a lot of the kits that you would normally use aren't compatible because we don't have large bead beating capabilities. We don't have a centrifuge. We can't use a lot of the buffers. So we had to kind of create our own optimized DNA extraction method. There are a lot of really great companies out there that are focusing on field-based DNA extraction methods that are efficient, but a lot of those are based for like more high biomass samples. So since we have such low biomass samples, we really needed to dig into some other methods. We've done a lot of work with the Claremont Biosolutions miniature bead beater because we really liked the idea of processing our swab samples with some bead beating. And then we've also done a lot with enzymatic extractions. And the last thing that we really had to work out was how are we going to purify our DNA and PCR products. So typically you would do a lot of centrifuge spin columns, but again, that wouldn't be able to work. So we've done some work with like some enzymatic reactions, and then eventually decided on bead-based purifications actually work really well in the spaceflight environment. So once we had a method that we felt really confident in, we wanted to be able to test it out in an analog environment because there's a really big difference between you as a scientist doing something in the lab and then giving it to some other crew member to do in an extreme environment. So we were able to train the crew before the mission, but it was a very limited, like about maybe 3 hours total of training on our hardware for the crew to actually implement it in the field. So we participated in NASA's Extreme Environment Mission Operations. For short, it's called NEMO. And it's a great ISS analog because it is located 5 miles off the coast of Key Largo, Florida. And it's at 62 feet depth. So it's secured onto the bottom of the ocean. And the crew members are completely isolated out there. They go and live out in this habitat for about a week or two. And it's a very small space. It can only fit 6 total crew members. They're totally saturated. They're under nitrogen saturation. So in order for them to leave the habitat, they have to undergo an oxygen decompression cycle. So they can't just leave the habitat whenever they feel like it. They're isolated out there, and they're kept very busy doing a bunch of extravehicular scuba diving training activities, and then they have a bunch of lab internal work activities too. So in our first field application, we had 6 different crew members collect swabs and run through our first version of our swab-to-sequencing experiment. And we got some really interesting results. We were able to get some return swabs. So we were able to do a comparison between MiSeq and Nanopore and found that they were comparable in the results that we were seeing. But the biggest thing that I took away from this first analog testing experiment was really the operational aspect of it. Like I said, it's really different to have a crew member having to recreate what you're telling them through just like a small set of procedures. And we really needed to work on operationally, how are we going to package all of our stuff and the logistics. For this experiment, we were using the Claremont Biosolutions bead beater, and we were using a larger swab. So it worked okay, but we really wanted to focus on optimizing the DNA extraction end. So we worked on that some more, and I'll talk about that throughout the rest of this presentation. But from what we were able to learn from participating at NEMO with some of the operational stuff, we were able to quickly transition part of the sample preparation to the ISS. So working with the Genes in Space program, we collaborated with them to combine our efforts and combine using the mini PCR and the MinION sequencer together. And in this experiment, you can see this picture on the left. Astronaut Dr. Peggy Whitson is collecting a little part of a colony from those microbial surface plates that we do for our microbial monitoring. So she was able to collect a colony and do a colony PCR reaction, and then prepare the sample using pipetting, just standard laboratory pipetting, and load it and run it in space. And then the culture sample was returned to the ground, where then I did our nominal sample preparation method that we do for normal return samples. So I did some Sanger sequencing and some Vitek. And the results that Peggy Whitson got on the space station matched the same results that I got on the ground. So this was really a groundbreaking first time that we've ever been able to identify microorganisms grown in space and identify them in space. And what I found most interesting from this experiment is that we could use pipetting, just standard laboratory pipettes. Before this, we weren't really sure what would happen doing liquid transferring in space. If we opened a tube, would the liquid just come out of the tube? But we found that surface tension really helped keep the liquid in the tubes, and that being able to just collect liquid at the bottom, you could just do standard pipetting. So from everything that we learned from our first analog experiment and the ISS application of Genes in Space 3, we went back to NEMO for a second mission and had a new and improved swab-to-sequencer protocol, which is what we're still currently using today. So we selected a new swab. So we wanted a swab that was a little bit smaller so that we could actually fit it into the DNA extraction. So we switched to an enzymatic-based extraction method where we were just using heat and enzymes to really optimize the yield from the swab. We've also had better Oxford rapid chemistry that we've been using, and updated our crew procedures and making it more streamlined. So in this analog test, we had 3 different crew members collect swabs from the environment. They collected from the phone, the swab, and the table. And our results were really promising, because they showed us that we found a lot of human-associated microbes, but also the majority of ocean-associated microbes, which makes sense because the habitat is located in the bottom of the ocean, and the crew members are constantly going in and out from their diving activities. So it makes sense that there would be a lot of ocean-associated microbes present. So now that we had a lot of the operational experiments sorted out, we wanted to just do a little bit more method comparison before we were ready to apply everything to the ISS. So we really dug into culture-dependent versus our culture-independent method. So we collected 2 swabs in tandem from the same location and put 1 swab into our nominal culturing method, where the swab went into some TSB. We plated it onto TSA and then isolated it to get pure cultures, then extracted that and amplified the first 500 bases of the 16S gene for Sanger sequencing. And then with our swab-to-sequencing culture-independent method, we extracted the DNA right from the swab, did the amplification of the full 16S gene, and then did the nanopore sequencing. So we had 51 total locations. There was 26 of them were from our Human Health and Performance Laboratory here at NASA Johnson Space Center, where we were able to swab a lot of analog equipment to what the crew members would be using on board for some of their exercising. And then we also swabbed 25 residential locations, so typical things that you would commonly be touching and using every day. And there were a total of 169 culturable bacteria isolates detected from those locations. So our results from this were really promising, from showing the comparison of the Sanger sequencing IDs of the culture versus the nanopore sequencing IDs from culture independent. The Sanger sequencing identified 96.5% of those cultures to the genus level and only 65% to the species level, but that is pretty common for Sanger sequencing. Our nanopore sequencing data detected parallel culturable organisms, but we also saw an increased diversity of other organisms present from our culture-independent method, which is not surprising because with our culturing method, we're really limited in what we're actually detecting. We're only selecting for microbes that can grow on one specific type of media at one specific temperature in aerobic conditions. So we know that our culture-independent data were able to detect organisms that are unculturable or really hard to culture and have specific requirements. We're also detecting microbes that might only grow in communities. And from this data, we did see that there were increased things that we normally wouldn't find in our culture methods. So we think that this culture-independent method is really supporting the potential to increase NASA's ability to do a more detailed risk assessment and how the microbes in the environment could be impacting the crew. So now we're finally ready to apply everything on the International Space Station. So we worked, this payload was called the Biomolecule Extraction and Sequencing Technology Payload. And we worked with astronaut Ricky Arnold and Serena Anand-Chancellor. And this picture here on the left, I love because you can see Ricky Arnold collecting his swab sample from the wall next to the dining table where the crew eat dinner, and they spend a lot of time just like around the table talking to each other. And he's swabbing this area right behind a spoon where you can see they just like store all their silverware attached to the wall. So they collected 3 different swabs from 3 different locations and put it through the whole swab-to-sequencer method and sequenced it on board. And our results from this were fantastic. So this picture is showing the first swabbing location where Ricky Arnold selected. And we found a lot of human oral associated microorganisms, which makes a lot of sense because if this is where they're eating and talking, you would find more oral associated microbes. The other two locations that we tested was the Japanese experiment module air return grid and the PMM curtain, which is a curtain that they put up to separate the PMM module from the Node 3 module. And the crew members actually do a fair amount of personal hygiene in the PMM. So from those two sample locations, we saw an increased abundance of more human-associated, like skin microbes, which would make a lot of sense for the activities that are happening there. And this data that we found aligned really nicely with the ISS microbial profiles that we have from all of our years of historical culture data, and also with previous molecular publications of other people having collected swabs and then returning them to the ground to do Illumina-based analysis. So all of this supports the idea that humans are really impacting the microbial population of the ISS. And we think that our method has really been a fundamental breakthrough in how we can do microbial monitoring in spaceflight and helping us move towards more culture-independent methods. The last bit of data I wanted to share from this is this Spearman correlation. I like that it shows that our swab-to-sequencer method can detect unique populations from different environments. So you can see that our NEMO samples group closer together because they have more of the ocean-associated microbes, versus the ISS samples in pink are grouped closely together, but more similar to some of the analog swabs on the ground that are similar to some of the ISS environments. So what is next for us? We have a lot of big goals that we still want to work on with microbial monitoring. We're currently working on the systems demonstration. It's called the Biomol facility. And we're doing side-by-side comparisons of our culture-independent swab-to-sequencer method at the same time as the crew members collecting our nominal culture-based samples. So we'll be doing that for the next year to be validating that more in flight. We also want to do a larger ISS nanopore microbiome study. So we have about 200 return swabs that we've been collecting that we want to do some more assessments of ISS microbiome. We want to then also incorporate quantification into our sequencing detection. So because the two parts of our acceptability limits are how much is there and who is it. So currently with the sequencer, we can really detect who is it, but we need to work on quantifying how much is there. We're also wanting to expand into fungal. So everything I've shown here is all bacterial, but we've been doing a lot of work on fungal identification with sequencing from the environment. And then additionally, we've been focusing specifically on surfaces right now, but we'll need to expand to water and air to be able to do the whole suite of our microbial monitoring, which we're also working a lot on in the lab right now, and hopefully going to be doing some demonstrations on orbit this year. And then finally, doing more in-flight data analysis so that the crew can actually get their results faster instead of returning data to the ground. But I just wanted to then also highlight that the MinION sequencer sequences more than just DNA. It can also sequence RNA and proteins. And so we really wanted to do some work getting into more RNA sequencing and also expanding our research beyond just looking at microorganisms in spaceflight. So we worked with some collaborators at the University of Santa Cruz in California to do some RNA sequencing in space. So with this experiment that we did, astronaut Ricky Arnold prepared an RNA sample for sequencing in flight, and I prepared a sample on the ground. And then again, we just compared how the results looked, and we saw that they were very comparable, and that the spaceflight environment didn't have an impact on the ability to prepare the RNA for sequencing. So there's still a lot of RNA sequencing work to be done in spaceflight, but this was just the first preliminary showing that it can work. So we're really excited that we have developed all of these capabilities on the space station. We have the mini PCR and the MinION. We have all the full suite of pipettes and consumables that we need to be able to do experiments. So we're excited about this opening up opportunities to other researchers to do more detailed molecular biology experiments in spaceflight. I know I'd be really interested in looking more at different gene expressions and seeing how those alter during spaceflight missions. So I just wanted to close out, go back to the beginning and just remind everyone that as we continue to explore wherever humans go, our microbes go with us. And then hopefully our MinION sequencers will continue to go as well so that we can continue to not only monitor the environment to keep the crew safe, but also explore to determine potential new life. So I just wanted to acknowledge a lot of my collaborators. So first of all, I have the Swab-to-Sequencing team. This was the original team that launched the MinION to the ISS. So there's Dr. Sarah Wallace, Dr. Aaron Burton, and Dr. Christian John. And our collaborators at the University of California, Santa Cruz that have helped us a lot, Dr. Mark Akinson, Dr. Matane Jain, and Dr. Benedict Patton. And my MinION team locally here at NASA, I thank them for a lot of the work that we've been doing currently, getting all this stuff up and running in our new experiments. I always have to thank my extreme environment molecular biologists. So all of the astronauts that have helped us complete this experiments on the ISS and in the NEMO habitats. And our large amount of collaborators with Oxford Nanopore Technologies, the Genes in Space mini PCR teams, and all of our integration support teams. It takes a lot of people to be able to do successful spaceflight experiments. So we're just really thankful to have such great teams helping us out. And yeah, I like to always close out my talks with some fun tweets from all the astronauts. I like to see that they enjoy doing the fun science activities as much as we do. So thank you guys very much.
Thank you, Sarah. Really, your talk was really interesting. And in fact, I'm seeing now some questions for you. So we can start with the first one. All of them seems to be anonymous at the moment. And I want to remember everyone that if someone wants to ask the question online, you just have to tell us and we will promote you as a panelist and for doing your question on live. Anyway, for the first one, for those samples that astronauts send to the lab on Earth, which conditions are used and how they can be kept stable during the whole travel until they reach the lab?
Yes, so what we currently do just for microbial monitoring, they're, like I said, they're grown on the TSA and the sabdextrose media, and they're just at room temperature. So they wrap them up. It's actually very interesting. They'll like wrap them up in tape to make them as small as possible. And they just get like shoved into the vehicle and returned from the ISS to the ground. So everything's just at room temperature. So when we get them back in the lab, they often have been like kind of crushed. So we'll have to then sort through and spend a lot of time isolating pure microbial samples. So there's not a whole lot that we can do from our culture plates to preserve them. But for other scientific experiments, they can put stuff in the fridges or the freezers. So if you've collected a sample that you want to preserve for like a molecular analysis, you can put it into the freezer and have it returned frozen to you.
Okay. Another question. Apart from microbial profiling, will spaceflight sequencing be applied to studying how space conditions affect microbial DNA in terms of mutations towards adaptation?
Yeah, so there's a lot of interest in doing that. Currently, since like our specific focus was just microbial profiling because that's what we do as an operational activity. But for our research, we've had some payloads where commonly people will like launch microbes to the space station, and then they'll grow them in the spaceflight environment and then freeze them and return them. And so there's been a lot of profiling done like that, or a lot of people doing ground-based analog experiments where they'll grow microbes in like a rotating bioreactor. So those are ways that we currently are looking at impacts of spaceflight environment on gene expression in microbes. But I know that there are some future payloads that are looking at doing some more whole genome sequencing of microbes actually in the spaceflight environment to detect those changes in the organisms. So hopefully a lot of that will be coming very soon. All of our work has just been basically like laying the groundwork for actually using this technology in space, but that's our goal, is that either work that we're doing or collaborators or other scientists get to do.
Okay. What is the weirdest sample, I'm not sure I pronounce it correctly, that astronauts have encountered and asked you about?
I mean, I like that one picture I showed of just like all that biofilm goop inside of the hose. Like that was just very strange to see. I think the thing that excited me the most was, and like we talk about the most, is that there was like a panel that had a fungal growth just all up and down. It was one of the pictures I showed on the left side. And that was because the crew members were storing their towels, like bungeeing their wet towels to the wall. And then all the water from that was going into the wall and forming just like this fungal growth. So that just further connects the need to include microbiology in engineering, which we do a lot of those testing on the ground to select different materials that are more microbially resistant.
Okay, the next question is from Peter Young. Most of your kit is designed for use on Earth. Have you had to make special equipment designed for space?
No, everything that we're using is all COTS off-the-shelf items. So that's what also makes it so easy, is that we can just use what other companies have made and then apply it to the space environment. So the things that make it more compatible for space is the like little tweaks and optimizations to the sample prep, but all the hardware is just COTS.
Okay. Mike Edwards want to ask, some bacteria produce oxygen. Could they get to space for producing oxygens in planets without atmosphere?
Potentially.
I guess so.
Yeah, no, there's a lot of, this is not my field, but we have a lot of collaborators who do a lot of more like astromicrobiology and are really interested in detecting new life. So there's some interest, and people are working towards taking the MinION sequencer and actually putting it onto like a rover and being able to do a more automated testing. So the cool thing about the MinION sequencer is that it doesn't have to be known DNA samples. The way that it works, you can actually sequence, like they call them exonases, so they're just unknown foreign nucleic acids. So that has a big potential to be used in more like astrobiology exploration applications. So who knows what we'll be able to find in the future.
Okay. Any other question? In the meanwhile, I have also a couple of curiosities now that I can talk with you. Most of the samples that you have been showing us are like mostly using the swabs on the surfaces or the water. Have you considered analyzing some samples from the crew? I mean, analyzing the flora, the microorganisms over the skin on the astronauts, or the using saliva or something like this?
Yes, so there's been actually several studies. There's a publication by Alex Voorhees about the astronaut microbiome. And they did a really big study of collecting swabs from the skin, the tongue, the mouth, the forehead. And they had some really interesting data showing basically how the microbial populations change during spaceflight, and which I found really interesting, that like they're different in spaceflight than they are on the ground. So there has been a lot of studies on that. We didn't do any with our sampling, but it is the idea that I like to think of is applying this towards medical operations. So if the crew member were to get some, like they commonly get like a skin irritation rash, just because if you think about it, in the spaceflight environment, like you're not shedding as much skin as you are on the ground because your clothes aren't like rubbing on your skin every day. So sometimes they get like a buildup of dead skin, which I think is what leads to like the microbial population changing. But anyways, I can see this application applying really easily to the crew has some kind of skin infection. Let's swab it and figure out what they are having. Do they have like a staph infection, or do they have like an acne? So then that will help us understand how can we treat the crew if they're feeling sick, because that's another thing compounding onto this is that like resupply of antibiotics as you go towards deep space, you need to be more careful about what you're treating and all of that. So I think there is a big application for crew health.
Okay, thank you. We have another question. If we find life in Mars or Venus or Titan, could this system recognize the unknown life form? So I'm guessing they are asking about metagenomics. They can be, you can use your technology to apply it for this?
Mm-hmm. Yeah, so that's what I was talking about, how the sequencer can actually sequence XNAs, so any kind of nucleic acids. The hardest part of the sample preparation is how you can extract your DNA or your RNA or your XNAs. So there's a lot of work currently being done about like how to optimize the sequencer end, and I feel very confident that the sequencer will be able to be used in these exploration methods, because a lot of people are working on like solid state nanopores. But the hardest part is going to be getting a DNA extraction method that can work. But there are a lot of people working on this in the future.
Okay, any other question? If not, I think that we will close this session. I just want to thank you, Sarah, for your time and your efforts answering all the questions from the attendees. Also, thank you from MDPI and the Genes Journal. Thank you very much.
Yeah, no, thank you so much. This is a wonderful opportunity.
Okay, so if everyone needs or wants to know more, please feel free to send us an email. And please visit the conference webpage during the week and next week for the next live session and for the edited video from Sarah. So bye-bye.
Thank you.